So this is part 2 of the acid-base buffer lab. And we're going to be filling in this chart and again talking about what a buffer is. A buffer is something that resists the pH change. So beaker number 1 had just water in it, and we were able to shift the pH very, very quickly because there was no buffer in it. Beaker number 2 was the buffered solution. So beaker number 1, this was unbuffered. And what we did is we took the initial color of the water and it was green. And the pH was somewhere between 5 and 6. Okay, then we added 3 drops of acid plus 3 drops of HCl at that point in time for our procedure. And it shifted it to a yellow-orange. And the pH was somewhere between 2 and 3. So 3 drops shifted it from a pH of between 5 and 6 to a pH of between 2 and 3. Just those 3 drops. There's enough acid falling apart, producing those hydronium ions, which are able to change the pH. Now, beaker number 2 was the buffered solution. This had some baking soda or sodium bicarbonate in it. And the initial color of this was purple. If you remember, we added it, we were a little bit off the charts. And we were trying to get it back to this final color using the pH probe. And, I'm sorry, this became a yellow color, and we tested that before with the pH probe, about 8.0. But this is the thing is we're going to be comparing it to is the drops of HCl. We added 20 drops of HCl versus adding 3 drops of HCl. So again, a buffer is able to resist the pH change. We added a base, and what that did is it was able to absorb all these particles of hydrochloric acid. So the buffer will act by allowing the solution to absorb a large amount of the hydrochloric acid before the color changes and the pH change is observed. So here is a question. What is a buffer system? Or more importantly, what is a buffer? And it's just this definition up here. Again, a buffer is, it's actually not just one molecule, but it's multiple molecules. So it's a system with molecules that resist the pH change, that resist, not stop a pH change, that resist a pH change. So I want to share with you what the reaction is within your bodies. Okay? We breathe in oxygen and we exhale carbon dioxide. Some of this carbon dioxide is actually used inside of our blood. And what it does is the carbon dioxide, let's do this reaction down below here. Actually, we'll start it up here. When you take your H2O and you add carbon dioxide, it produces this molecule called carbonic acid, H2CO3. And this is what's used inside of our blood. This molecule can actually fall apart, and when it falls apart, it produces H plus and HCO3 minus. However, we can recombine that back together to form the H2CO3. And we'll come back to this in just a minute. You can also take this H2CO3, and if we bring in a base, which is an OH minus, if we bring in a base with this, it splits this molecule up, and it forms H2O and HCO3 minus. You will not have to memorize these, but I want to show you what's happening. These are reversible reactions. So what happens is this is sitting in our blood, and this is also sitting in our blood. These two molecules act as our buffer system. However, if we increase the acid, so this is Le Chatelier's principle, if we start to increase the acid, for whatever reason, let's say the chemical reactions inside of our body are producing more acids than they should be. Well, what happens is we have this molecule that's present inside your blood to absorb that excess acid. Just like the baking soda up here was able to absorb all the excess acid. It took, again, 20 drops to get to the same pH where it took 3 in an unbuffered solution. When your buffer is present, you're able to absorb this extra acids. Let's take a look down here. This molecule is also present, and if the base starts to increase, we have the other molecule that's present. So when these two molecules react, it's going to be forming water, which is neutral, but it's also going to be forming this compound here as well. So take a look at the end product. We're absorbing the acid, we're creating something that is neutral. We've bound up that H+. This molecule is taking the OH-, and it's turning it into water, which is also neutral. So our buffers have an effect of neutralizing either our acids or our bases.